Isolation of oral stem cells for tissue and organ regeneration. What does it really mean? The surface of the mouth and the gums contain special type of cells, fibroblasts. Among these cells, there is a subpopulation of even more unique progenitor cells called stem cells. Stem cells offer great promise for new medical treatments, since they can be guided into becoming specific cells that can be used in people to regenerate and repair tissues that have been damaged or affected by disease. However, extracts of oral fibroblasts may contain heterogeneous mixtures of different cells and only a tiny proportion of stem cell population, which is so crucial for regenerative therapies. So, we need those stem cells, but how can we identify and isolate them? Well, it can be done in 5 stages. First, one needs to extract a tissue from the gums with minimally invasive surgical procedure. Then, one can identify specific molecules on each subpopulation of the cells in the extracted tissue using a genetic sequencing technology. The molecules can be used as markers to distinguish these cells, just like an ID for every cell. The next step would be to use those markers to isolate the stem cell population with special antibodies that emit fluorescent light when attached to the markers on the stem cells. Once isolated, those stem cells can be used to demonstrate the superior potential towards bone forming to regenerate the skeletal tissue. The final product will be a set of specific biomarkers which allows isolation of cells with regeneration potential from the oral mucosa-derived fibroblasts. It is important to note that the experiments described in stages 1 and 2 are routinely performed in our lab. Our research already identified markers for isolation of cells with significant differentiation potential. However, proof of concept is required to accurately isolate the cells and demonstrate their superiority. Therefore, we ask for financial support.